Okay, so our last speaker for this session this afternoon is Dr. James Womack from the Department of Texas A&M University. He holds a title of Distinguished Professor at Texas A&M and is the W.P. Lewis Professor of Pathobiology in the College of Veterinary Medicine. He's also a member of the Faculty of Genetics and holds a joint academic appointment with the Department of Medical Biochemistry and Genetics. His degrees are from Abilene Christian University and Oregon State University, and he's served as a coordinator for the NRSP 8 project, which is a complementary one to NRSP 9. And he's president of International Society for Animal Genetics and American Genetics Association. And he's a member of the National Academy of Sciences and a fellow in the American Association for the Advancement of Science. And he's received a number of degrees and is a very prolific publisher in his career as well. So with that, I'd like to introduce Dr. Womack. His title of his talk is Nutrigenomic Applications for Improving Livestock Sustainability.
Hey, thank you very much. It's a pleasure to be here. I've learned a good bit about nutrition today, and I am talking about nutrigenomic applications for improving livestock sustainability. I'm a geneticist. I do genomics. I would be presumptive for me to suggest experiments that you might do with genomic technologies. But what I can do, I think, is refresh you or update you on some of the genetic and genomic technologies that are now available for our livestock species, and maybe have some discussion on how these might be applied to some of the phenotypes that I've heard described today, which have to do with sustainability, increased efficiency of foodstuffs, et cetera. So when asked to talk about this subject, the first thing I had to do was to learn what is nutrigenomics. And I found a number of definitions of this term. Most of them somewhat similar to this, and that is the study of the effect of genetic variation on dietary response and the role of nutrients and bioactive food compounds in gene expression. So there are two components here. One is, how does genetic variability of animals affect the way they respond to particular diets? And the other is, how do diets alter the expression of genes in individual animals? Both of these, I think, have a tremendous potential for impacting our use on genomic technologies and future nutritional studies. And both of these refer to the animal itself and its genes. I will not talk about other very important aspects of genomics, and that is the microbiome, which is another set of, if we look at the gut, ruminants, maybe 1,000 species. And another very important aspect of genomics is the feed stuff. How about the genetics or the genomics of the plants that these animals are eating? So nutrigenomics could have a very broad, a very wide scope. But I'll concentrate today on the pigs and cattle and chickens themselves rather than their microflora or on the plants that we feed them. I want to first talk about genetic variation, primarily because this is an area of my own research, and how this might affect how the variability in the genomes of individuals might be exploited in improving the nutritional status of animals, whether it be domestic or global. There are two types of variation that are extremely important. One of these we refer to as SNPs. SNPs are SNPs, single nucleotide polymorphisms. And one of the most important things of the many genome sequencing projects that have been done, which now includes most of our livestock species, is the discovery of the enormous number of nucleotide variations that exist in our DNA. We as humans have 2.7 billion of these base pairs, ATs and GCs, in our genome. Most mammals have about exactly the same, cattle and pigs. Chickens have less than half that number. What's interesting and what we learned is that along these DNA chains, approximately 1 in 150 or so of these nucleotides pairs varies between us, or varies in a population, has the potential to vary between us. So on these 2.7 billion base pairs of DNA in a cow or a pig, there may be 20 million genetic variants. Now, these are scattered along the genome. Many, as you probably know, 2% or less of our genome is actually in the exons that code for products, but that's not to say that the rest of the genome is not important. We used to talk about junk DNA, and now that we're learning that the folding of DNA is very important, and it's very difficult for us to say that these intergenetic regions are not important. In fact, we know that a lot of the regions that we once called junk DNA are very highly conserved between mouse and human and cows and pigs, and that conservation certainly suggests a function. Now, these SNPs, or these variants in our DNA, can now be tested very efficiently and fairly economically with the chips shown on the upper right. The human, some 1 million of these SNPs have now been put on microarrays, so any one of us could, for $150, could find out our repertoire of these SNPs along our genomes. We have a cattle chip that has about 800,000 of these SNPs, somewhat evenly spaced across all the chromosomes, so we can easily test individual cows, pigs, chickens, with these microarrays that tell us which of these variable nucleotides vary in a specific individual. Another type of genetic variation that's becoming very important to look at is copy number variation. And these CNVs, or copy number variants, are illustrated at the bottom of the slide. This picture is of human amylase genes. And copy number variation is in amylase genes in humans is very important in how humans metabolize starches. Some of us have more amylase genes than others. There's no difference in our amylase genes. We just vary in how many of these we have. And it turns out that maybe 25% of our genome exists in these regions of copy number variation. And this also can be tested very effectively. The bottom right is shows a little stretch of a genome in which hybridization reveals the relative amount of DNA at a particular location. The reference individual is the baseline in the middle, and individuals that have increased numbers and copies of this DNA get the blip up, and individuals that have less copies of a particular DNA than the standard are the blip down. And about 25% of our genome exists in one of these blips. So we differ in the number of copies that we have for a lot of our genes. So how can we utilize this SNP technology? We are beginning to utilize it very effectively, I believe, in studies that we call GWAS, or Genome-Wide Association Studies. This is an illustration of a human study which is pretty much standard. I think there are some 1,000 reports now of GWAS analyses in humans. We have a number of these now coming forth in cattle and other livestock species as well. Basically, this utilizes the SNP chip. So if you can find a large number of individuals that are cases for whatever phenotype you want to describe, and a similar number of cohorts that you can call controls for that phenotype, you can test every one of these individuals with one of these SNP chips. And then if you have a particular genomic region that is important in that phenotype, you should see an association of one of the alleles of that SNP with the case as contrasted to the controls. And this is very much a fishing expedition. It's not hypothesis-driven research, and yet it's extremely important for the first identification of some of the genomic regions and even some of the candidate genes that underlie specific traits. It's especially effective for complex traits where we do not have a single gene controlling 95% of the variability of the trait. We have effectively used GWAS to identify genes that contribute to 2 to 7 or 8% of the total variability in a polygenic trait. So very quickly, let me just tell you a study that we have going. This is bovine respiratory disease. We knew in the beginning that the heritability of susceptibility to bovine respiratory disease is significant, and therefore we assume that we would have some potential to identify genomic regions that contribute to susceptibility. This is one of the NIFA CAP projects that I have the pleasure of directing. About 6 different institutions are involved. This is a dairy calf farm in Tulare, California, in which thousands of dairy calves are raised in these crates, and some 10% of these calves will develop a bovine respiratory disease. The cowboys go through here not on horses but on golf carts, and they identify sick calves. We have a scoring system Sheila McGurk developed. We identify a score, and then we pick an animal in a nearby crate that's perfectly healthy, and that becomes the control cohort. We've put together some 1,500 cases and 1,500 controls from this study, and then we have done the association. Now, there are several different methods you can do associations, and as you might guess, if you're looking at 800,000 SNP markers, you're going to get some false positives. We put fairly high criteria for statistical association. And then you analyze these, a variety of methods, and you get something like is shown at the bottom. This is called a Manhattan plot because the results look something like the skyline of Manhattan. If you get statistically significant associations, each of those peaks, each of the colors represents a different cattle chromosome. And starting with the big chromosome 1 on the right, going down to the smaller ones. This, of course, with 800,000 markers is highly squeezed together. But those peaks represent particular SNPs that were highly associated with the disease. And we now have 30 or 40 very highly significant markers in regions where we've identified candidate genes and are going back and sequencing animals to get the validity of these genes. If you don't have any response, you don't get a Manhattan plot, you get a College Station, Texas plot, in which the skyline is just kind of like this. But fortunately, we have a pretty good Manhattan plot here. I want to say that our goal is to present producers, and we're also doing this with beef cattle from feedlots. We don't have the feedlot data analyzed yet. We're still collecting. But and we probably will get quite different peaks. It's very unlikely that calves just a few days old utilize the same genes in combating pathogens as adult feedlot cattle do. So we're anticipating that we'll see some different genes. Jerry Taylor at the University of Missouri is the director of another CAP that is doing the same type of study for feed efficiency, in which a number of feedlots are contributing data in which individual efficiency of utilization of feed is measured, and they are also finding hits. And so we're beginning to accumulate a set of SNPs that identify these important markers, these important traits. We want to produce a small chip, which any producer can buy for $20 or $30 with just a few hundred markers on it. But each of those markers has a known significance to the phenotype. And of course, the goal is to produce, in our case, is to produce cattle, breed cattle, that are resistant to BRD, to reduce the needs for antibiotics as much as possible, and consequently contribute to production in that way. But these techniques are available to just about any phenotype that you can imagine, for which you can produce a set of cases or a set of phenotypes and a set of controls. Certainly these have limitations. It has to be a heritable trait. The number of genes, the magnitude of effects. We're probably going to end up with several dozen genes for BRD, each contributing to maybe 2 or 3 or 4% of the total variability. And then you have to have the animal populations. And this is sometimes pretty hard to come up with the animal resources necessary to conduct experiments of this type. The other side of that definition of nutrigenomics was the role of nutrients on gene expression. Certainly technologies are available to look at expression of genes. We have expression arrays in which you can look at the different cDNAs produced on a microarray. These, of course, are specific to tissues. We now can look at the RNA directly, RNA-seq, more expensive, but also extremely informative on how particular nutrients affect the expression of genes. Even as far back as when I took basic genetics, was taught that a phenotype is a result of genes and environment. I can't think of any environment that more likely to affect a phenotype than nutrition, because the nutrients and products of nutrients are directly in contact with the genes. The tissues in which the genes are expressed are in direct contact with metabolites which have the potential to affect expression. We're just now opening the doors into epigenetics, where this is simply where identical genomes don't necessarily produce the same phenotype because of usually some type of methylation which occurs in the DNA, some type of effect on the histone modifications on the chromosomes, and this whole area is just opening up, and I think is going to be extremely important in nutrition research. This can be fairly expensive. There are some limitations, and I think a major limitation is the fundamental information for hypothesis testing. What nutrients do you want to look at to see how they affect gene expression? And some basic research has to provide this fundamental knowledge. Another aspect is biotechnology and genetically modified food animals or genetically modified crops and foodstuffs. Certainly we've had transgenics around for years. We can now make knockouts in cattle and pigs, where we actually can knock out a gene instead of just randomly putting genes into the genome. Thanks to cloning technologies, which allows us to utilize the somatic cells in tissue culture and identify our knockouts, and then utilize those somatic cells to clone animals. We now know a lot about, we know that a lot of RNAs exist that have the potential to knock down gene expression. Certainly these have potential. And as you all know, there are many limitations to this type of research. Not only is it expensive for cows and pigs, there are regulatory limitations or public perception issues that have to be addressed. But still, this is an area where modifications of genomes may have some significant impact on future research in nutrition. With those remarks, I would gladly yield to our panel discussion and see if some questions might come from the audience.